Hi, today we're going to take a look at the beginnings of the neurobiology of behavior. And in this topic, we're going to be looking at biology and behavior, the basic cells of the nervous system, how two neurons communicate, and finally, learning and synaptic plasticity. And first, the one on the list is, we'll have a bit of an overview first. And in this overview of this is an overview of the whole neurobiology section of the course. We're going to take a look at the big question, which is who and why we are, or how we can find out about that. We're going to look at the aim of this topic. We're going to look at the role of the brain in human individuality. We're going to look at neurons, the regulation of brain and behavior, ethology, and finally an example of how it all fits together. So for thousands of years, human beings have pondered who we are and where did we come from. And they've come up with some weird and wonderful explanations, none of them based on reality, none of them based on evidence, all based on human thought. And finally, we're able to put some reality or evidence into the situation. And that is going to completely change how we look at ourselves, how we consider ourselves. And the behavioral biology is one of the best sciences available for doing this. And it's working on understanding who and why we are, using biology as its foundation. Now, the aim of this topic is just the whole neurobiology topic is to examine how our brains regulate our thoughts, emotions, and feelings, how our brains make us individuals, the individuals that we are, and how our brains are regulated by evolution, genes, experience, and hormones. So it's not all down to genes, things involved as well. Now, the role of the brain in all of that is to channel or sort all the inputs that come from the environment, from our genes, from our experience, and from any other sources possible, and process all of that information and turn it into behavior of some sort. So it's the final pathway by which our individuality is shaped from biology and experience. Now we're going to take a look at neurons, and we'll start with the function of the nervous system at the level of the single neuron, which is like the basic cell of the brain. And eventually we'll examine how millions of neurons in a particular region of the brain operate as a network, how they speak to each other. And we'll focus on the regions of the brain most pertinent to emotion and behavior, rather than bodily functions. In fact, most of the brain is devoted to behavior, and only a small part is devoted to bodily functions. So we're going to look at regulation of the brain and behavior and how that's done. We'll cover how the brain regulates hormones and how hormones influence its function and the behavior of our own behavior. We'll examine how natural selection has shaped our behavior and how it is mediated by brain function. And we'll focus on the bridge between evolution and the brain, namely the genes, and how they evolved. So we're going to be looking at evolution and how that fits into the brain, how it's through the genes and into the brain. So evolution created the genes, and the genes create the brain. Now we're going to take a look at a thing called ethology, which is studying animal behavior in the natural environment. So we're going to take a look at the whole thing, everything that makes up the human behavior, from hormones, evolution, genes, and the environment, and how all of these affect each other. The environment affects all of these things, and all of these things affect the environment. And that is the study of ethology. And that's an example of how it all comes together once we've gone through all the preliminary bits. We're going to look at the behavior of aggression. And one of the main reasons that we are going to do that is because it's been well studied. There's lots of information about how it works in the brain and what leads up to it, and what are the consequences of it. And it's, of course, a very important subject in amongst the human species. We're going to be looking at, well, now we're going to look at biology and behavior. And this is just an introduction. It's a bit like getting us pointed in the right direction. We won't be covering a lot of it in this particular section, just enough to set us in the right direction. So in this section, we're going to be looking at the how the study of neurobiology and evolutionary biology is an interdisciplinary approach. It's not just one discipline. And we're going to be looking at the biology of behavior. We're going to be looking at categorical thinking, how that works. We're going to be looking at non-categorical thinking when it comes to thinking about human behavior. We're going to look at a research blueprint. I mean, in other words, how we go about researching human behavior from a neurological point of view. We're going to look at the context of the research, and we're going to travel from neurons to networks. So in other words, we're going to start with neurons and finally end up with networks in the brain. And we're going to look at how individual differences occur over time. And we're going to be looking at some of the factors affecting behavior. Now, with an interdisciplinary approach, excuse me, the purpose of this topic is to explain the biology of what makes us who we are, and particularly as far as becoming individuals, in other words, the differences between people. And then moving on, we're going to look at the biology of behavior, which we're interested in how biology fits into the picture of human behavior. It's a new approach in terms of for over 100 years, biology wasn't considered as being relevant to human behavior. And finally, we've decided to put it back into the equation. We've decided to include biology in the study of behavior and individuality. And we're going to look at the fact that can changes in behavior occur as a result of the mutation of a single gene. And from our previous studies, we've seen how easy it is for genes to mutate when cells are dividing and the DNA information has been copied. So there's a biology to our sexual choices, and to the extent and type of our religiosity and everything else about us. So in other words, biology affects just about everything we do as human beings. There's nothing that's outside the realm of biology. So we sometimes we approach the study of human behavior using categorical thinking. In other words, lumping behaviors and people into categories and then trying to work out what it all means. And the reason we do this is because it helps our memory, easy to remember things if they're in categories, but it distorts our abilities to see differences. And we have trouble seeing the big picture. So biological sciences, or the evolutionary biological sciences and neurobiology, prefers to use non-categorical thinking. In other words, accepting that when we're thinking about behavior, rather than trying to lump people into categories, recognize that everybody is different in terms of how their brains work, how their brains are constructed, what genetic information they have, and how this affects their interaction with the environment and the environment's interaction with them. In other words, it's not a single factor involved. So that's one of the reasons why we use an interdisciplinary approach, because the study of behavior involves lots of aspects of human functioning. Now, the blueprint we use for the research is we ask where the behavior came from. In other words, how did it start? And we study the brain and the nervous system, so we know how that works. And we work back in time to the things that modulate the nervous system, such as environmental triggers, hormones, and perinatal and fetal development. We look at genetic attributes, that's like the physical attributes that are genetic. And we examine ancient natural selection pressures that led to these genetic attributes. In other words, that's evolutionary natural selection through evolution. So all of those things will be considered in the research process. And also we need to take into account the research context. So we're looking at everything from things that happen in a matter of milliseconds in the brain to evolution that happened over 6 million years. So there's a wide scale to cover there. We'll start out by looking at neurons. So we'll look at a single brain cell, the neuron. Then we'll look at how the neurons communicate with each other. We'll look at large networks of neurons. And we'll look at the nervous system and how it regulates our cells. So we're starting small and ending up big. But that's not all today. That'll be covered over a series of topics over a number of weeks. Now, individual differences can change over time. In other words, what you are today may not be what you're going to be in 20 years' time. In other words, because of mutations and experience and environmental changes, how we respond and how we behave is changing all the time. So it's a bit like the philosopher, the old Greek philosopher, who said, you can't step into the same river twice. And the reason for that is that the river's flowing past, and it's a different river every millisecond. So if you step into it now and then you step into it a second later, it's a different body of water. It's not the same water that you were stepped into before. So human beings are much the same. We're changing all the time. And we call that process of change plasticity. And of course, the factors that affect behavior are the environment, hormones, early life experience, fetal life, in other words, even before we're born, genetics, and evolution. So it's sort of starting with evolution, then the evolution created genes. Genes affect how the baby develops in the womb. Then we have our early life experiences. Then our genes regulate our hormones, and the environment is the genes are triggered on or off by the environment. So it's all together, all works together. That's from genetics to the environment. Now we're going to look at the basic cells of the nervous system. And first we'll mention that the human species has been evolving for 6 million years. So we've had quite a long time for our brains to take the shape that they have and the form that they have. We'll look at the basic building blocks of neurobiology. And we'll start with the neuron and glial cells. and we'll look at electrical activity in the brain, and we'll look at behavioral individuality. So if you want to understand human behavior, know where it's coming from, the biological basis of it, we need to consider that we have been evolving for 6 million years. So that's a lot of time for changes to occur, for the species to take the shape it has. And it hasn't been a straight line, as we've seen before, that there have been quite a few hominin species, particularly over the last 2 million years. And however, we didn't evolve for 2 million years. We have been evolving for 6 million years. It's just that we became hominins about 2 million years ago. And of course, Homo sapiens has only been around for about 150,000 years. So our mob is quite recent compared to the 6 million. Now, the basic building blocks of biology, we'll start with an overview of the neuron, what that's about, and its various parts, and how each part functions, and how it communicates with other neurons. Now, the basic neural constituents, like in the main brain, is the brain itself is the neuron, and holding the neurons together, like the glue in the brain, are glial cells, G-L-I-A-L, glial cells. And looking at now the brain, or the neuron, consists of a number of parts. We have the dendrites, which are like the ears of the neuron. That's where the signals come in. That goes to the cell body. In the middle of the cell body is a nucleus. Then out of the cell body, we have a long tail. Now, just before the beginning of that tail is a thing called the axon hillock, and the tail is called the axon. Excuse me. And then that long axon, that tail, leads down to can't read that one there. It leads down to the part which connects with the next or it's called the axon terminal, which connects with the next neuron, and it connects via a thing called a synapse, which is the gap between the one neuron and the next neuron, or the beginning of one neuron and the end of the end of one neuron, the beginning of the next neuron. Now, the ears are called the dendrites, and they stretch out in one direction, or actually in just about multiple directions, and they connect with other neurons, and the messages get passed from neuron to neuron to neuron. And the chemicals that excite the dendrites cause electrical impulses in the neuron, and that's where the work happens. So in the cell body is where the energy is produced. So a wave of chemical excitation passes from the dendrite through to the cell body and down the long cable of the axon. The neuron cell body integrates all the inputs of all the different neurons around it. So you've got a whole bunch of neurons around a particular neuron, and all the inputs from those neurons through the bunch of dendrites is integrated by the cell body and then passed on to the next cells through the axon. I'll be giving you nice pictures of these, so if the explanation doesn't make much sense, the verbal explanation doesn't make much sense, the pictures will. I mentioned the axon hillock. That's just before the tail, or the axon, at the end where it leaves the cell body, and it's the axon hillock that that passes the electrical signal from the cell body to the axon, and it has quite an important function in maintaining the signal, or it could sending the signal and maintaining the signal. The axon is, of course, the long tail, and that goes down to the axon terminal, which connects with the next bunch of dendrites in the adjacent neurons. And that's where the electrical signal is passed. But when it gets to the synapse, or the end of the axon terminal, then that's where the electrical energy stops, and it has to be converted into chemical energy. Now, the electrical activity of the brain enables the communication within the cell, and it consists of resting electric potentials and action electric potentials. Now, while the cell is supposedly resting, it's actually working quite hard, because it's the cell body and the axon hillock are monitoring all the signals that are coming in and determining whether to pass them on or not. And there are some basic rules as to what enables a signal to be passed on and what doesn't. And the axon hillock actually makes that decision as to what's going to be passed on or not. So this requires, even though it's so-called resting, it requires a lot of energy to do this sorting out of what the incoming signals, and whether or not to send them on. Yeah. Now, information coming in is called neuronal input, and so it comes in by a single dendritic spine, and it turns into ions, and ions are positively or negatively charged atoms, and they begin to move, and they go down through the cell body. But a single neuronal input won't trigger an action potential. In other words, it won't turn the neuron on, as it were, and send the signal to another neuron. It has to be sufficient neuronal input for that to occur. A single one won't do it. And the axon hillock won't produce the power needed to pass the message down the long tail of the axon to the axon terminal. Now, the way that the action potential is triggered is two ways. It's temporal summation and spatial summation. Temporal summation means that you get the same input coming in over and over again. So it's like a single signal, but many, many times over. That's over a period of time. That's temporal summation. So that could trigger action in the neuron. Or spatial summation, which could be a number of different messages coming in all at the same time. And that also might signal the axon hillock to send the message down the spine. So when there's enough wave to reach the axon hillock, the axon hillock integrates the various inputs and decides whether or not to act. When the axon hillock is triggered to act, the neurons are in action potentials. In other words, if the axon hillock isn't passing the message on, then it's in resting potential. If it is passing the message on down the axon to the axon terminal, then it's in action potential. Now, the action potential doesn't reduce over space and time. In other words, even though it's going down the long tail, the level of the electrical signal is maintained, and time, like, it doesn't decay over time either. It keeps regenerating, and all this is done through the axon hillock. It regenerates the signal. It sort of amplifies it, keeps it going until it's time to turn off. Now, in a mature brain, in other words, after about 25 years of age in a human being, The axons, that's the long tails with like electrical wires, have a insulation sheath wrapped around them. But before that, it's in the process of having the insulation put on, but it's not completed until after about 25 years of age. And the meaning of that is that that until the axon sheaths are completely insulated, then axons don't work very fast, and they can also short circuit with other axons. So you can get a lot of random signals happening, and a lot of that's why like young adults or teenagers can be such wild creatures. One of the reasons is that their axons aren't insulated yet, and signals are flashing all over through their brain. And not only that, some thinking processes are a lot slower because it needs the insulation to speed up the speed of the signal going down the axon. Now, this sheath is created by glial cells, and I've got a nice diagram of that in the PowerPoint slides, so you don't have to worry too much about that now. So over a period of time, the axon becomes completely sheathed, and then it's working at its full effort. It's working at its top speed, and it won't short circuit with other axons. And like I said, we don't achieve full brain function until after 25 years of age, which is a bit of a worry because the digital age is actually created by people who are under 25 years of age. So the digital age has been created by people whose brains aren't fully mature yet. A frightening thought. But like all the other blips in human evolution, we'll deal with this one just like we dealt with all the others. Okay. Now, behavioral identity, individuality, neurons are complex and networks are complex, and the way and the mutations in genes occur quite frequently, and that affects how the structure of the brain and the functioning of neurons. So there's no two people that are exactly the same in behavior, even though we like to categorize people and put criminals in that box and white-collar criminals in that box and blue-collar criminals in that box and depressives over here and anxious people over there. In fact, it doesn't work that way. We're all a mix of all sorts of strange things, and you really can't get an exact fit for anyone into any single category. And if you ever see the Diagnostic Statistical Manual of the American Psychological Society, and you see the huge number of symptoms that are possible for any given mental disorder, then you begin to wonder. And the criteria is a sort of a certain percentage of these symptoms have to be present, which means that a whole lot of others won't be present. So that makes me wonder and say, well, that sort of tells me that it really isn't a category, because you've got a huge number of different symptoms, and people may or may not have them and still fit into the category. So it's like categorizing people against all the evidence saying that there's just too many things that can happen with human behavior as far as people being individual and unique. But because we need to fit them into, or shoehorn them into boxes, into categories, then we're going to do it by this sort of shonky method of creating a long list of possible symptoms for any given disorder, and say, well, if they've only got 25% of these, then they qualify to fit into this label. For me, that's not a very good qualification at all. Okay. Okay, moving on to the next section. So we're going to talk about how two neurons communicate. And we're going to talk about the synapse, which is the gap between the end of one neuron and the beginning of another. We're going to talk about neurotransmitters, which is the chemicals that exist in this gap that conduct the signals from one neuron to another, but they do it chemically, not electrically. Now we're going to look at the effects of drugs on these chemicals and the effect of these chemicals on individuality. And I remember when I was starting out over 40 years ago as a new social worker and learning psychotherapy, there was a big war going on between the psychiatrists who said that mental disorders are all the cause of a chemical imbalance in the brain, and the others were saying, no, it's all down to learning. We learn it from our parents. Our parents drive us crazy or something like that. And if you belonged to one school, you couldn't belong to the other. So it was a big sort of what do you call it? Rivalry between the two ways of seeing the world. And over the years, I'm beginning to see that actually it's not two ways. They both work. It is chemical and it is environment. It is learning and it is chemical. Both of them are happening at the same time, and they interact with each other. So, looking at how two neurons communicate. Well, in this particular topic, we're going to look at the nervous system and how it produces behavior, and how everything on the planet that came from the nervous system, that came before the nervous system, regulates its function. So in other words, the nervous system we have now is the end product of everything that went on this planet before that. In terms, I mean, the things that affected the human species, of course, and our parents, our grandparents, our great-grandparents. So the DNA that you carry in you and I carry in me has filtered out through environmental disasters, some people not surviving and others surviving. And we are the end product. We are the survivors. We are the children of the survivors. Or you might actually say, well, we are the children of the people, the winners. We are the children of the people who managed to knock all the other members of the species on the head, so that they couldn't survive. Yeah, not a nice thought when you think about it. It's also going to look at how information moves across the synapse from one neuron to the next. And we're going to look at how electrical signals are changed to chemical messages in the synapse. And this is a critical foundation for understanding how the brain works, and how drugs are used, and the effect of drugs and how they're used to influence the chemicals. So now we're going to talk about the synapse, which is the gap between one neuron and the next one. It's a very tiny one, and the electrical signal stops at the end of the axon terminal in the previous neuron, and won't start again until it's chemically crossed the gap, the synaptic gap, to the dendrite of the next neuron along. So the message from one neuron must be translated into a different language to cross the gap, and then passed to another neuron, and then passed on. Now we've got some nice pictures of a synapse, how it works. So you don't have to think too hard now. And we'll talk about the neurotransmitters that conduct the signal from one neuron to the next. So like I said, the electrical signal in the preceding axon creates the chemical that's going to cross the synapse. And these chemicals or neurotransmitters are contained in vesicles in the axon terminal. So at the end of the axon, the little fingers at the end of the axon, each one right at the end have vesicles containing these chemicals. And there are different chemicals for different purposes. So it's not one chemical fits all. They're all quite different. And these chemicals have particular receptors. So it's like the receptor has to fit. And if the receptor doesn't fit, then that chemical isn't going to pass the message on. It's a bit like a virus. A virus can only bind to the cell if the receptors on the cell match the corona on the virus. So the shape of the neurotransmitter and its receptor must be complementary. In other words, they must fit together. It's a bit like a lock and a key, or key and a lock. And if the key doesn't fit the lock, then the message won't be passed. So the binding of the neurotransmitter to its receptor changes the excitability of the next neuron. So once that binding occurs, then the next neuron starts to get excited. It starts to light up. And multiple receptors exist for the same neurotransmitter. So in other words, it's not quite simple. It's a complicated picture. Now, neurotransmitters, once they've done their job, can deactivate themselves, and they can either be recycled back into the vesicles, or on the previous synapse, previous axon, or they can just decay. They can be broken down by enzymes. So either of the two can happen. So there are multiple types of neurotransmitters. Not all of them are excitatory. And the same neurotransmitter can have multiple purposes, multiple uses. Okay. Now we can have inhibitory transmitters, and we can have excitable ones. So it can work both ways. You can have a neurotransmitter that turns the next neuron on, or it can have a neurotransmitter that turns the next neuron off. Okay. Now, neurotransmitters are made up of very cheap and basic chemicals that are available in the body. So they're made up of simple amino acids that you can get out of almost any human diet. So it's not a what do you call it, a highly rarefied sort of chemical. It's a very basic body or physiological chemical, amino acid. Now these neurotransmitters are produced quickly. It doesn't take long to make. And they can be squeezed out. Many messages can be squeezed out of a single pathway in the vesicle. So it's not as if it's one vesicle, one neurotransmitter. And of course, the neurotransmitters are easily recyclable. Now, to determine whether a chemical is a neurotransmitter, it must be located in the axon terminal. That's its sort of home, and it's released during the action potential. So that is sort of the rule, the golden rule. If it doesn't do that, then it's not a neurotransmitter. It's something else. And I forgot to mention that sheath, or the insulation around the axon, is actually made by glial cells. And glial cells act a bit like glue, but in this case, they're acting in a way that enables a build-up of this insulation over time on the axons. And a neurotransmitter floats across the synapse and receptors on the postsynaptic neuron. So that's another condition. And any interference with the neurotransmitter will alter the neurochemical events in a predictable manner. So in other words, they're the requirements. You've got it's located on the axon terminal. It's released during action potential. It crosses the synapse to the receptors and the postsynaptic neuron. And any interference with the neurotransmitter will alter neurochemical events in a predictable way. Now, the effect of drugs. Some drugs block the access of the neurotransmitter to its receptor, in other words, prevent it from binding, and stopping the communication across the synapse. For example, curare, in the movies and adventure books, the South American Indians put on their poison darts, poison arrows, can block the acetylcholine receptor in the diaphragm. So that's in the chest, and it will, by blocking that receptor, the message that keeps the chest pumping, keeps the chest going, stops. So the chest will no longer pump air. It will no longer take air in and expel carbon dioxide out. And of course, the person will suffocate. And it suffocates by the receptors in the diaphragm being blocked, or being sort of, the receptors not being available to the appropriate neurotransmitter hormone. Okay. Now, antipsychotic drugs can block the dopamine receptor, and dopamine, of course, is mostly the pleasure chemical in the brain. However, if you block the dopamine receptor, it can also lessen the symptoms of schizophrenia. So if you have schizophrenia and you block that receptor, then the symptoms, we call it, are ameliorated. And drugs that can also break down the recycling of neurotransmitters, or they can completely destroy a neurotransmitter. So the idea of the drugs is to interfere with neurotransmitters to produce certain effects. So if you destroy the neurotransmitter norepinephrine, it will lower the blood pressure in the body. So if you want to have lower blood pressure, take a pill that destroys this particular neurotransmitter, and that's the neurotransmitter that makes us anxious and stressful, among other things. So that by doing that, we're no longer anxious and stressful, and our blood pressure goes down. Mind you, it means that if there's a time comes when we need to be anxious and stressful, then we're in trouble. Now, antidepressants cause amplification of the neurotransmitter signal by blocking its degradation. So antidepressants keep the signal going long past when it would normally decay. And I must add here that the human species evolved, the brain evolved, so that any pleasures that we get are short-lived. In other words, you cannot have a pleasure that's going to last forever. No, it doesn't happen. It doesn't exist. The reason for that is that our DNA wants us to keep doing pleasurable things, so we will produce more DNA and pass it on to the next generation. Now, if you did it once and that was all you needed, and that pleasure, that happiness stayed with you forever, then you'd cease doing what was necessary to pass your DNA on to the next generation. So in order for us to keep going, keep trying, and to produce as much DNA and pass it on as possible, then we need to need a lot of repeating of the pleasures that we have. And of course, the more we repeat a pleasure, the more we need to do it to get the same buzz. In other words, over time, the pleasure goes down. And the reason for that is not because nature is nasty. It's because our DNA wants to be passed on to the next generation and wants it to do all the things that are necessary to do that. And that usually involves some sort of pleasure. Now, the manipulation of chemicals always has the risk of side effects, primarily because each neurotransmitter has multiple jobs. in different parts of the brain. So if you destroy a particular neurotransmitter that's doing a job in one part, then it will also stop it from doing a different job in a different part of the brain, which may be bearable or may be unbearable. And I guess people who make these drugs have to keep that in mind. Now, neurochemistry has a lot to do with our individuality. And if we look at the neurotransmitter endogenous benzodiazepine, they influence our anxiety levels. So if we have a problem with producing and releasing benzodiazepine in our brain, our anxiety levels will go up. If we have sufficient benzodiazepine and we produce enough of it, then our anxiety levels will go down. So it's whether you're anxious or not doesn't depend upon how you were treated as a baby, not entirely. It also has a lot to do with a particular neurotransmitter in your brain, whether you're producing it well or not well. However, having said that, we have to bear in mind that our DNA is switched on and switched off by the environment. So for our DNA, our genes to do their job, certain environmental things have to happen in order for them to work. And if those things don't happen, then those genes don't work. For example, the mothering instinct. Young girls don't, or even women in general, I believe, don't walk around all the time feeling the strong mothering instinct that you have when you've got a baby in your arms. You need the environmental influence of looking down or holding a very small baby in order to trigger that neural network in the brain, or they turn on those genes. And without that, it's not turned on. I mean, there are other genes at work, of course, the need to produce babies, to make babies. That's a different need. That's not the same as mothering. The actual mothering bit only starts when you look or hold a small baby. If you look at chooks, although chooks can get clucky, they don't actually need to have a baby, but there's something in the environment, in a chook's environment, from time to time that switches on the clucky bit. If that wasn't there, then the chook wouldn't get clucky, and that's probably the side of eggs, but not all side of eggs causes cluckiness. So it's a constant interaction or interplay between the environment and our genes that makes us do what we do. Okay, now the next section is learning and synaptic plasticity. Means synaptic plasticity means changeability in the synapses. And we'll take a brief look at the cortex and the hippocampus, its role in learning, and then learning itself. So the cortex and the hippocampus, if you haven't found out already, are highly involved in memory. And of course, if you're learning something, it involves memory, mostly. You need to remember what you're learning. So the parts of the brain that involve learning, involve memory, are very important for learning. And one of the problems, if you have problems in the hippocampus, it's possible that you could develop Alzheimer's. Elderly people, of course, their hippocampus shrinks with time. But if it shrinks too fast, too soon, then you can get Alzheimer's, where you've virtually lost your memory. Not in a total way, but in a very, very serious way. Now, some people believe that learning is actually a change in the strength of synaptic communication. In other words, if you can achieve strong synaptic communication, that's communication from one neuron to the next, if that can be increased in some way, then that will improve the learning process. In other words, you will learn better. Which, in other words, it's really making new pathways in the brain. So it's to excuse me, to learn, you need to make new pathways, which involves synapses. So in a long time ago, it was believed that whenever something new was learned, a new neuron or new synapse was formed, and then that went out of fashion. But now it's coming back again. It actually is showing with our modern techniques of research in the brain, we're beginning to see that it is true, that learning creates new synapses and new neurons, new pathways in the brain. In fact, as we'll learn later, it's not so much the neurons, it's the connections that do the trick. It's a bit like people. We do better if we've got good connections with other people. And neurons do a lot better if they've got good connections with other neurons. They don't work very well on their own. Okay. Right, so that's it for today. I hope that was instructive. I'll be sending you the PowerPoint, and that will contain a lot of diagrams, which will help in understanding this lesson. Okay, till next time.